Everybody, welcome to Biotech Academy. First of all, wishing you all a very healthy and happy new year. I would like to apologize to all my subscribers for not being able to upload any videos from long time, like it's almost it's today's 67th day when I uploaded my last video, and trust me, because of certain unavoidable circumstances I was not able to do so. But now let's start with a fresh, and we'll continue with where we left, and I'll try to upload videos very frequently so that you can just go through them and you can prepare for your June net. And so let's start with myoglobin. We've discussed about different types of protein structures, different levels of organization, and we were discussing about some of the globular and globular proteins. So let's discuss about hemoglobin today. So as the word suggests, hemoglobin, it is a hemoprotein where heme is the prosthetic group, and it's a conjugated iron containing, hemoglobin is an iron containing conjugated protein. And just to remind you, what is conjugated proteins? Conjugated proteins are proteins where we have a prosthetic group and a protein component, and this prosthetic group, it is non-proteinaceous in nature. It may be like different elements, but it's non-proteinaceous in nature. And what is non-proteinaceous in nature means it is not composed of any amino acid. And this protein part, it is a polypeptide, or it is a linear chain of the 20 standard amino acids, different combinations of the amino acids. So in hemoglobin, heme is the prosthetic group. And what is heme? It is the base is the porphyrin ring, and this acts as the prosthetic group. Alright, so we'll discuss about the structure, the functions of these in this hemoglobin protein in detail now. So we'll continue with this. And so hemoglobin, where it is found? It is found in the erythrocytes, or it is found in the RBCs, or the red blood cells, or they are also called as erythrocytes. And it is made up of four peptide chains. So it is a tetramer. Basically, four peptide chains are present in four peptide chains are present in the hemoglobin, which are all these four peptide chains, they are connected via non-covalent interactions. All right, so these peptide chains, they are connected via non-covalent interactions. And porphyrin ring forms the base structure for the heme. Heme is the prosthetic group. So what is the structure of this porphyrin looks like? It is okay, and here we have N, here we have NH, here we have N, here we have NH, and then we have double bonds and at various places. So it's here, then here, then here, here, not this place. So this is the basic structure of the porphyrin ring. And now iron. So heme contains Fe, and this Fe exists in two forms, Fe2 plus and Fe3 plus. So the porphyrin ring, the protoporphyrin 9, which has Fe2 plus, it is ferrihemoglobin, and it is ferrohemoglobin. Fe3 plus, it is ferrohemoglobin. And then the oxygen, what is the function? You must have studied about it in your lower classes, that the function of hemoglobin is to transport oxygen to the different parts of the body. So hemoglobin, where iron is in Fe2 plus form, then only it will bind to the oxygen. If it exists in Fe3 plus, then that hemoglobin does not have the affinity for oxygen and won't be able to bind to the oxygen. Now let's come to the various types of hemoglobin which is present in human body. Alright, so there are different types of hemoglobin are present in the body. And how do they differ? They differ in the peptide chains. So I've already mentioned that this hemoglobin, it is composed of 4 peptide chains, and these 4 peptide chains, they vary in different types of hemoglobin. So in adult human body we have HbA1, hemoglobin A1. It is the major part, and it is of the 98% of the hemoglobin which is present in the adult human body. It is of the HbA1 type. And what is it composed of? It is composed of 2 alpha chains and 2 beta chains. So we can also write it as alpha 2, beta 2. Alright, so this alpha chain, it is composed of 141 amino acids, and this beta chain, it is composed of 146 amino acid residues. So this is the majority, this is the major component of the adult hemoglobin, which is of HbA1 type. And the remaining 2% is HbA2 type. That accounts for the remaining 2% of the human adult hemoglobin. And this HbA2, it contains 2 alpha chains and 2 delta chains. So this delta chain, the alpha chain is very similar to the one which is present in HbA1, but then this delta chain, it is almost similar to the beta chain, but the difference between this alpha and the delta chains are of 5 amino acid residues. So the beta chain and alpha chain are the same in both HbA1 and HbA2, whereas the other 2 chains, which is beta and delta chains, they are almost identical, but they are almost same, but only differences of 5 amino acid residues, which makes it beta and this one delta. Alright, so however, this is about adult hemoglobin, but then the hemoglobin which is present during embryonic and fetal developmental stages, they are different from the ones which are found in the adult human body. So during embryonic stages and during embryonic and fetal developmental stages, different sets of hemoglobins are present, and the one type of hemoglobin which is present in the early embryonic state, it contains zeta chains. Alpha chains are again same, however it contains zeta chains which are most similar to the alpha chains, and 2 epsilon chains which are similar to beta chain. So what is the difference between the adult hemoglobin and the fetal hemoglobin which is present in the early embryonic stage? So in adult hemoglobin we have alpha beta or alpha delta chains, whereas in embryonic developmental stages, we have zeta and zeta and epsilon chains. That is the difference. So, and these differences are due to the composition of the amino acid residue. Now coming to the iron. So porphyrin ring in Heme is So porphyrin ring in Heme is protoporphyrin 9, and this iron exists in, I've already mentioned, it exists in Fe2 plus and Fe3, though in 2 different oxidation states. One is in Fe2 plus and the other is Fe3 plus. So, and this is ferrohemoglobin. Ferro, the one with Fe2 plus is ferrohemoglobin, and Fe3 plus it is ferrihemoglobin or methemoglobin, or it is also known as methemoglobin. And this Fe when it is in Fe2 plus oxidation state, then this one has affinity for oxygen, and so hemoglobin with Fe2 plus can only bind to the oxygen and can transport this oxygen to different parts of the body. Alright, so what are the functions? So there are some of the functions of hemoglobin. What are the functions of hemoglobin? It is the functions of hemoglobin as first and foremost function of hemoglobin is in the transport of oxygen. Then second is transport of carbon dioxide. These are the two major functions of hemoglobin. First is transport of oxygen from heart to different parts of the body, and then transport of carbon dioxide from tissues from different parts of the body towards the heart for the gaseous exchange. And then it has other important function is it acts as buffer, biological buffer. So it is a biological buffer. Hemoglobin is a biological buffer, and we'll discuss how it acts as buffer. And it also transports nitric oxide. So NO, the ferrous oxygen binding, the oxygen binding site, which is because of the ferrous ion which binds to the oxygen, that also has an affinity for NO, and therefore it also facilitates in the transportation of nitric oxide, NO. And this is where, in which part of the peptide chain this affinity is there? It is present in the beta chain of the hemoglobin molecule. Alright, so these are some of the functions of hemoglobin, and then we'll discuss about how it binds to and leads to the transportation of oxygen. So there are certain reactions which occur inside the body for this transportation of hemoglobin. So there are various reactions which occur between hemoglobin and oxygen. Alright, so the reaction between oxygen and hemoglobin, please make a note of it, that although addition of oxygen to any molecule is oxidation, however the reaction which occurs between hemoglobin and oxygen, that is not an oxidation reaction, it is an oxygenation reaction. So it is oxygenation of hemoglobin and not oxidation. Please, it is a very important point that it is oxygenation and not oxidation. So there are various steps. There are series of steps which occur during the oxygenation of hemoglobin, and hemoglobin actually reacts with 4 molecules. Each hemoglobin molecule reacts with 4 molecules of oxygen and forms Hb4O8, and it transports oxygen in this form, Hb4O8. So the sequence of the reactions are like: first, hemoglobin, 1 molecule of hemoglobin, Hb4, binds to oxygen and it forms Hb4 and O2. Then this Hb4O2 combines with another oxygen molecule and forms Hb4O4. Then again this Hb4O4 binds with another molecule of oxygen and forms Hb4O6, and in the end this Hb4O6 binds to another molecule of oxygen and forms Hb4O8, and then it is transported, the oxygen is transported with hemoglobin in the form of Hb4O8. Okay, so now it is does this hemoglobin transports oxygen, and how does it has affinity for specifically for oxygen? So we've already please recall when we were discussing about the tertiary and quaternary structure of the proteins, I mentioned that it is the tertiary or the quaternary structure of the protein which determines its function, and actual or accuracy in the protein folding for tertiary and quaternary structure. So accuracy in the folding is very essential for the protein to perform its function. Any deviation from any deviation or any fault in the protein folding may lead to an incorrect folded protein, and thus the protein loses its function. So it is the quaternary structure of the hemoglobin. It is the quaternary structure of the hemoglobin which is essential and which determines the function of the hemoglobin, and it is the quaternary structure of the protein which has the affinity for oxygen, and the oxygen molecule, it binds to oxygen, and the oxygenation of hemoglobin is because of the tertiary or the quaternary structure of the hemoglobin molecule. So the take home message is that the quaternary structure of the hemoglobin is essential, and it is the quaternary structure which has the affinity for oxygen. Now in deoxyhemoglobin, so there are two different states. There are two different states in which hemoglobin molecule exists. One is the T or the tensed state, and the other is the R or the relaxed state. So the hemoglobin molecule, it exists in these two forms. Alright, so what happens in the oxygenated form and what happens in the deoxygenated form? In deoxygenated form, when the hemoglobin is deoxygenated, so the globin units of the hemoglobin molecule, they are tightly bound and it exists in the tensed state. Okay, and this tensed state, because of this tight closed binding between the globin, because of the closed binding between the globin, because of the closed binding between these globin units, this tensed state has reduced affinity for oxygen. However, when any one of the four globin units, when one binds to the oxygen, then this tensed state, it is converted into the relaxed state, and it is the relaxed state binding of one oxygen molecule facilitates the binding of the other by increasing by enhancing the affinity. So what happens? It is cooperative binding, means binding of we've already discussed when we when we were discussing about the reactions between oxygen and hemoglobin to form Hb4O8. So there were a series of 4 steps. So when the first oxygen molecule binds to this hemoglobin, and it is converted into the relaxed state. In the relaxed state, binding of one oxygen, it facilitates it further enhances the affinity of this relaxed state hemoglobin for the other oxygen molecule. So it becomes O4, Hb4O4. So, and then Hb4O4, it again increases the affinity for Hb4O6. So it is cooperative binding, means binding of one molecule further enhances the affinity for the same molecule. So that is cooperativity. Okay, so it is the tensed, the oxygenated and the deoxygenated forms of hemoglobin. It is because of the tensed and the relaxed state, and it is the interconversion of the tensed and the relaxed state leads to the oxygenated and the deoxygenated form, and interconversion of these two forms only facilitates in the transportation. So the hemoglobin transports oxygen in the relaxed state, and then during the gaseous exchange between oxygen and carbon dioxide, because during the gaseous exchange, then again the relaxed state is converted into the tensed form, so the affinity for oxygen is lost and the oxygen molecules are released. So and then it binds to the carbon dioxide, so it gets deoxygenated basically. So so I hope it is clear how this tensed and relaxed state facilitates oxygenation of hemoglobin and its transport. Now if we plot a graph for the transportation of oxygen by this hemoglobin molecule, then we get a sigmoidal curve. What kind of a curve we get? We get a sigmoidal curve, and it is like this, where on the x-axis, if we plot partial pressure of oxygen, which is the unit is millimeter mmHg, and here it is percent saturation of hemoglobin. So if it's like 0, 10, 20, 30, 40, 50, 60, okay, then 10, 20, 50, 60, 70. Okay, so it's this kind of a sigmoidal curve is obtained, which shows the cooperative binding and the saturation curve of the hemoglobin molecule. So this is percent saturation of hemoglobin, and this is partial pressure of oxygen, and this is the curve which we obtain if we plot a graph and under ideal conditions. But then this varies, and this varies under different conditions, like there is an effect of pH, there is an effect of temperature, and also because of it is affected by the concentration of, it is affected by pH, then it is affected by temperature, and it is also affected by concentration of BPG, which is 1,3-bisphosphoglycerate. So these are the factors which affect the transport of oxygen by hemoglobin. So these are the three factors which we'll discuss now. So what happens if the pH or the temperature is altered on oxygen transport? So the first is pH and temperature. What is the effect of pH and temperature on transport of oxygen? So an increase in temperature. So if we increase the temperature, an increase in temperature or a decline in pH, an increase in temperature or a decline in pH, how does it affect the transport? Then this curve, this sigmoidal curve which we drew just now, this sigmoidal curve, it shifts towards right. Means in this situation, higher partial pressure of oxygen or the higher oxygen concentration is required for the hemoglobin to get saturated and carry out the transport. So an increase in temperature or a decline in pH, it leads to the shifting of this curve towards right, so that here if we are plotting here partial pressure of oxygen and it is percent saturation of hemoglobin, so the curve shifts towards right. So what happens? For the saturation to attain a higher partial pressure of oxygen is required. Alright. Similarly, if however the reverse happens if there is a decline in temperature. So if there is a decrease in temperature or an increase in pH, under both these two circumstances, this shift towards left. Means under lesser partial pressure of oxygen, this saturation will be attained. So these are the two factors which affect the transport or the transport of oxygen, and these are the factors which affect the cooperative binding of oxygen and hemoglobin molecules. Then the third is what we'll discuss is the if concentration of, how does this concentration of BPG, or it is also alternatively it is known as 1,3-bis phosphoglycerate. Okay, so 1,3-bis phosphoglycerate, how does this concentration affects the cooperative binding and oxygen transport in together with hemoglobin. So what happens is that BPG, it is, what is it? It is a highly charged anion. It is a negative anion, and which it binds to the beta chain. So BPG has affinity for beta, or you can say that beta chain has an affinity for BPG, which is a negatively this BPG is a negatively charged anion, and beta chain of the hemoglobin, beta chain of the deoxy, actually the deoxy hemoglobin has the affinity for BPG and it binds to BPG. So what happens? HbO2, if there is a high concentration of BPG, then it is this oxygen in the hemoglobin, it is replaced by BPG and we get Hb13BPG and oxygen molecule is released. So there is oxygen molecule is released if in the presence of this bisphosphoglycerate. So BPG, what it does, it reduces the affinity between the hemoglobin and oxygen and it replaces the oxygen so that this oxygen is freed from the hemoglobin. And in this equilibrium, what it does, more BPG will bind to more of hemoglobin and more oxygen is liberated. All right, so then that is why the in the fetal hemoglobin has more affinity for oxygen, and so this fetal hemoglobin has more affinity for oxygen than the adult hemoglobin, and that is why the oxygen during the development, during the gestation period, the oxygen, how does the fetus gets oxygen? It gets oxygen from the mother. So what happens? This fetal hemoglobin, because it has more affinity for oxygen than adult hemoglobin, so that's how the transport of oxygen occurs from the mother's blood to the fetus. And then the other function is the transport of carbon dioxide, so which occurs in the tensed state of the hemoglobin. So these are some of the important points of hemoglobin which you all must know from the exam point of view. And that's all for today. If you have any doubts and queries from this topic, you can always ask in the comment section or on the email ID. I'll be happy to answer all your queries. And once again, I apologize for not being able to upload videos, but I'll try to upload more videos so that greater amount of syllabus is covered in short duration. And we'll also, in the next video, we'll discuss about some of the questions. We'll discuss about myoglobin in the next lecture. Along with that, we'll also discuss the previous year's questions from these two topics, like hemoglobin and myoglobin, which have been asked in this GATE, NET and DBT, GRF, etc. So that's all for today. Wish you all the best and work hard. Thank you.